Hi guys, I think we ended up stopping last week on Hardy-Weinberg equilibrium, so we're going to go ahead and get started in the middle of this chapter on Hardy-Weinberg equilibrium. Like we talked about last week, Hardy-Weinberg equilibrium is just a method that people use to kind of measure allele frequencies in populations where they might not really know what's going on. Like I said, this is a kind of a theoretical model. It doesn't really work very well on extant populations because there are some stipulations for Hardy-Weinberg that usually make it difficult for an actual real life population to stick to. But it is used for guesstimating allele frequencies, and it is useful theoretically for things like endangered species and trying to figure out allele frequencies in a population at one time, especially if there is a deleterious or bad allele floating around and you need to figure out what the frequency of that is. So if you are trying to figure out what percentage of the population might have these alleles and what percentage of them might be able to pass those on to their offspring, this is kind of a useful thing, especially, like I said, for endangered species or species that are in danger of becoming extinct or endangered. So the Hardy-Weinberg equilibrium consists of two variables, the P and the Q. So the P is just the frequency of the dominant allele, or the one that will be shown if you're talking about just Mendelian genetics, if you have two genes, one dominant and one recessive. So an example that we used in class, like cystic fibrosis. And so the normal allele for cystic fibrosis would be dominant. The Q is the frequency of the recessive allele, or how many of those alleles there are in that population. So for our cystic fibrosis example, Q would be the number of actual cystic fibrosis genes that are floating around in the population. And so as you would probably infer, P plus Q, so all the dominant alleles and all of the recessive alleles added up in a population have to equal 1, or 100%. So that's going to account for all of the alleles that are in that population at that time for that specific gene. So you can expand this equation out a little bit, and you can actually use this to map genotypes as well. And so you can predict how common a genotype is using the expanded equation P squared plus 2PQ plus Q squared equals 1. I know that looks a little bit like a lot of math, but it's not really. So if you consider that P, again, is the dominant allele, P squared is going to be the homozygous dominant genotypes. And so that's going to be like big A, or big S, those homozygous dominant genotypes. 2PQ, because it's got one of each of those alleles, is going to be your heterozygous individuals, and then the Q squared is going to be the frequency of the homozygous recessive genotypes, and so like the little a kind of genotypes. And so where you've got two recessive genotypes or two recessive alleles at the same time, those individuals would show a recessive phenotype if they were to get those two alleles from both their mother and their father. And so again, don't think too hard about this. I know it looks like a lot of math, but it's not really. It's just kind of placeholders for the different types of genotypes you might see in a population. And so in a parent generation, we're using these little cute little kangaroo rats right here. Oh my God, they're so cute. So if you're looking at a population, and they've got a pretty good sample size here. So we've got 1,000 kangaroo rats, and there's 3 different phenotypes here. So we've got a dark brown kangaroo rat. Genotype is the big B, so this is the dominant phenotype, and so this would be represented by your P squared in your Hardy-Weinberg. A spotted kangaroo rat, which is the heterozygous individual, so it's got one dominant allele, one recessive allele, and that gives you kind of this mottledy looking kangaroo rat. That one's my favorite. This one would be represented by the 2PQ in your equation. And the light brown, this is the recessive phenotype. And so this is going to be little b. This is going to be represented by Q squared in your population. And so for this population specifically, it's got the number of individuals down here. It looks like that dark brown phenotype is probably selected against in this population just because you have a very low number of individuals here. So we've got 16 out of 1,000 show this phenotype. And so this is probably, like I said, selected against. Imagine if you had dark brown kangaroo rats, maybe on a sandy, beautiful light brown background. These guys would probably show up more, which means they're going to get picked off by the hungry hawks and the foxes or whatever that are hanging around. This guy, there's a few more, 222 out of your 1,000, so about 22%. So this is a little bit more common, but again, maybe these ones don't blend in as well because we've got the recessive phenotype is 76% of our population, 76.2% to be exact. So what you've got here is a lot more individuals, and so for whatever reason, this is probably the most common phenotype because it's probably selected for in that environment. Like I said, I imagine a dark or like a light sandy kind of desert environment. That's where kangaroo rats have come from. You find these in the desert Southwest, and so probably having that light brown color is going to be more advantageous in that environment. So the way that you calculate the allele frequencies for P, you've got 2 times 16, so we've got 16 individuals. That's where this number comes from. And then 2 because you've got 2 of each of these alleles, okay? So again, don't get confused and don't think too hard about this. So that 2, just because there's 2 here, 2 big B's, times 16 because we've got 16 individuals. And then we're going to add that to 222 because there's 222 individuals that are heterozygous, meaning that there's 222 of these big B alleles floating around just in these individuals that have this phenotype. And so if you were to put those together and add them up, divide them by 2,000, 2,000 is just the total number of alleles in this population. We have 1,000 individuals, okay? Each of them have 2 alleles each, and so 2 times 1,000 is 2,000. That's where that number comes from. So if you divide this all out, you end up with 0.127. So this is 12.7% of the population have at least one of these big B's in their genotype, okay? We're figuring out Q down here, 762 right here, times 2 because we've got 2 of these in the population, plus 222 because we've still got our heterozygous individuals here. 222 of them have one of these. Divide by 2,000 equals 0.873. So that means 87.3% of this population has at least one recessive allele in its genotype. And so that's where those numbers came from. So those are the allele frequencies. Frequency, if you think frequency, think percent. Because we want to know how many are in this population at this time. That's what the frequency is. So for Hardy-Weinberg to work, one of the rules that we have to abide by is that there has to be random mating. And again, this is one of those things that's difficult in a real population to find because usually there's some sort of sexual selection or somebody's being picky about who they're mating with. And so random mating is not usually something that you see very often in a natural environment. But again, we have to assume this for Hardy-Weinberg to work. And so when you look at your Punnett square up here from the numbers that we got before, again, our allele frequency for the dominant allele is 0.127, and then the allele frequency for the little allele, the recessive allele, is 0.873, okay? And again, these should add up to 1. P plus Q should equal 1, if you remember the first equation. So if you set these up in a Punnett square, so we've got our adult individual here, this is dad, and dad is a heterozygous male, and we've got mom up here, and she's a heterozygous female. And so depending on how their eggs get together and sperm get together, this is going to be the percentage that you're going to get for a heterozygous dominant individual. So we do 0.127 times 0.127 is 0.016, so 1.6% of this population, or of the births from these parents, is going to be a heterozygous dominant individual, okay? That's where that number comes from. PQ is just P times Q, so 0.127 times 0.873 is 0.111, and then there's 2 of them, that's the 2 PQ here. And then Q squared is just the 0.873 times 0.873 is 0.762, that's where you get those from. And so the genotype frequencies from those numbers are going to be P squared, 0.016, 2PQ is 0.222. Remember, there's 2 of them, so we have to add them together. And then the little alleles here, the recessive alleles, are going to be 0.762. And so that's where those numbers came from, um, down here. So if we have the next generation of rats, if we had um, our heterozygous individuals mating together and we already know what our allele frequencies are, this is the genotype frequencies that you would expect to see in this population. So again, here's those rules and the assumptions that we make for Hardy-Weinberg that make it a little bit difficult to find in a real live population out in the wild. But again, this does work theoretically and it is very helpful. So it applies when there is no natural selection, so there's no pressures working on anybody. So there's not one phenotype that is more selected for than another. No mutations, because that's going to jack up all of our allele frequencies if we just throw a new one in there because of random mutation. No migration, meaning that nobody is coming or leaving. We're not having any of our alleles leaving the population just because they decided, like, screw this population, I'm going somewhere else and starting my own colony. None of that's going on. No genetic drift, meaning that we've got no individuals coming in from other populations bringing in new sets of alleles. And then there also has to be random mating. So nobody can get picky about who they're mating with. We can't just only get with the super hottest, greatest male, and we're not just going for the biggest, fattest females that look like they can produce the most eggs. That's not what we're doing here. It has to be totally random. Just, hey, are you up for this? I'm up for this. Sweet, let's make some babies. That's what's going on. So, there are things that cause some individuals to have more offspring. And so this is, again, that survival of the fittest that we talked about before. And so individuals that have sets of traits that make them either better at hiding, better at finding food, better at getting mates, they're going to be the ones that have more offspring, because if they hide better, that means they're going to live longer, they'll have more chances to mate. If they are really good looking and they look really good as far as sexual selection goes, those ones are going to get picked more for mating and they're going to have more babies. If they're better at finding resources, they're fed more, they have more energy, they're going to have more energy to devote to mating rather than just foraging for food. And so that fittest, that's an actual term, and fitness is a measure, like an actual physical measure of the reproductive output, i.e. how many offspring we have, of an individual with a particular phenotype, so what they look like on the outside, relative to the reproductive success of others of the same species with alternative phenotypes. So, for example, with those little kangaroo rats, the ones that were more blonde in color must have had more fitness than the darker brown ones, because you see more of them in the population. And so those ones must have been selected for in some form or fashion. And so they're having more babies. And because more of the light brown mice are having babies, then the darker brown ones are getting picked off, or they're not having as many babies. Then you start seeing a shift in how many of those kangaroo rats are available in a population at that time, depending on what those selective pressures are that are working on that population. So things that are important to fitness are traits that confer the highest fitness are things that include genotypes or phenotypes in the population that are better than others in the population. And so if your genotype includes, in the kangaroo rat example, little alleles, so we've got our recessive alleles, and even if you're a heterozygous individual, that gives you more of an advantage than having both dominant alleles. And so an individual's fitness is measured relative to the other genotypes in the population. And so all of this has to be measured against other phenotypes. So if everybody looks the same, we can't really measure what the phenotype, or which phenotype is better or more fit, because if everybody looks the same, then what are we measuring? So there has to be some variation in the population for us to be able to measure this, because one of those phenotypes or one of those phenotypes is going to be better than the others. And so that's how we're able to measure that reproductive output. Oh, there's a kangaroo around my gosh, so cute. So fitness also depends on the environment where the organism lives. So like we were talking about before with the kangaroo rats, if you've got blonde rats that are on like light blonde sand, these guys are going to blend in better, and so they're probably not going to get picked off as much by predators because they can hide better, especially when you've got Bob over here who's got dark brown fur. And so this guy's going to get seen really easily, and so he's probably going to get picked off by the rattlesnake first, if you get what I'm saying. But again, if you are in a environment where you're against a dark brown background, this is where this guy is going to get picked off more easily. And so all of this depends on the environment where the organism is. And you can start seeing directional selection that favors one extreme phenotype when you've got a shift in environment like this, because you have to have the correct phenotype for the correct environment in order for that to confer some sort of advantage to you. So you can see what's going on with the allele frequencies down here as well. And so if we've got about half and half, and after a few rounds of hawks picking us off, pretty soon you're going to see what we saw in the other example, where you've got like 87% recessive alleles and then only like 16% of those dark alleles. So fitness also depends on the organism's reproductive success compared to everybody else. And so if everybody has one baby, like emperor penguins do, that doesn't really give us much to measure. But if you've got kangaroo rats who can have, 3 or 4 litters a year, if you've got certain females that have really good genes and they have really good phenotypes, and they're just like selected for in that environment, their reproductive success is probably going to be higher. That's just a measure of how many babies that little guy can pop out during a season or during its lifetime. And so that's going to be the reproductive success. So some animals do need to adapt to new environments, like we were talking about in class with the bird box analogy. Adapting is just when organisms become better matched to their environment, either because of random chance or because they're able to think through that new environment very quickly, and they're able to adapt to it better. And so if it's a matter of thought, or whether it's a matter of just chance, these are things that need to happen in order for organisms to survive, because especially in a rapidly changing environment, organisms need to be able to change quickly in order to match that environment, so that way they can continue to take advantage of resources and then also continue to mate and have offspring. So one of the things they talk about in your book is that natural selection is not goal oriented, and so natural selection doesn't work on populations with a goal in mind. That being said, natural selection also does not make perfect organisms, and so what happens with natural selection is just that those that are the best adapted to their environment at that time are usually going to be the ones that have more babies, send their genes into the future, into their offspring, and then we're going to see more of those genes and more of those phenotypes come out in future generations. So with the Galapagos finches, there was a really cool study that was done. It started in like the early 1970s and went on through the mid 80s. You guys can see here what these biologists did was they went out and they actually measured rainfall, and they measured beak size, depth specifically. And so the depth is a measure of from the nostrils all the way down to the bottom of the beak right here. And so they measured depth of the finch's beaks right here. And they noticed that during dry years, what happened is that the beak size in millimeters went up. And so during really dry years, what the finches had to work with as far as food was these really hard, large seeds. And so having a bigger beak depth, which gave them more torque to crack nuts and crack seeds, was advantageous in that time. And so the ones that had the bigger beaks, like this, the ones that had the deeper beaks like this, did better. And so what they saw was an uptick in how big beaks were, and that's because the ones that had the bigger beaks had more access to food. If they had more access to food, they could spend more time working on mating and making babies rather than foraging for food. And so the ones that had the bigger beaks were the ones that gave their offspring those genes. During wet years, however, the finches, there was a big influx of these really small, soft, probably more like a perennial seed here. And so the smaller beaked birds were selected for during years where they had a lot of rain. And that's because you've got this really big fat like honker right here. It might be really good at cracking these kind of nuts, but it's not so good at like stabbing little tiny itty bitty baby seeds out of the grass. Whereas a bird that had a smaller, more spear-like beak, that's going to be a better phenotype for that type of resource and that type of food. And so during wet years, what they saw was that average beak depth went down, and they started to look more like these little um, spear-shaped beaks rather than these big old honker like parrot beaks up here. And so this is just the ability, just in the course of just a few generations, again, because this is from what, like maybe 1975 up to like 1985, only 10 years. And so what they saw was pretty big fluctuations in the size of the beaks just in a few generations. And so these genes that are working and these phenotypes that are working can be cyclical, and the ones that are best suited during that year are the ones that are going to pass their genes down. And again, there's several phenotypes here. There's one phenotype here, one phenotype here, and we're measuring the success against each other. And so it's relative to the different phenotypes and the different genotypes that we're looking at. So we can't make a perfect individual. And again, this has to do with the fact that natural selection is not goal oriented, and so these pressures that are working on these populations don't have an end goal in mind. It's not like the environment is like, oh, I think I'm going to rain a lot this year because I really want those finches to have really sharp beaks in 50 years. That's not what's going on here. So this is a day by day, year by year iteration that we're looking at. It's not something that's goal oriented. And so things that stop a population from progressing towards perfection is the fact that environments change. And so what might be perfect for one environment, dry year, is not going to be perfect for a wet year. And so because the environments change so quickly, we can't have one phenotype that fits both of those resource methodologies. And so we can't have a deep-beaked and also a short-beaked bird at the same time. And so environments change very quickly. Variation is needed because that's the material that natural selection and the environment are working with to kind of mold those populations into working the best in that environment. And there might be multiple alleles for a trait, which causes individuals to have the same fitness. And so that's also a factor here. We talked a little bit about artificial selection before, and so I'm going to go over this again. Artificial selection is just when humans are in charge of breeding plants, bacteria, animals for desired traits. And so I think like the best example of this obviously is dogs, because dogs all used to be very similar. They all were kind of wolf-looking creatures, and we've bred them down so hard that now we have apple-headed chihuahuas, which you guys have probably all seen before because they're adorable, to like German shepherds, and to like Alaskan Malamutes and Great Danes, and all different kinds of dogs, like, you know, hundreds and hundreds of different breeds of dogs. So artificial selection is when we as humans are acting as the selective pressure, because we are selectively breeding organisms together that are going to give us exaggerated versions of a desired trait. So like a boxer dog, for example, you like them to have that really smushed brachiocephalic face. We like them to have those really long arms, that really deep chest. They're good at running, and they're really good family dogs. We breed for personality traits as well as physical traits. So one of the things they talk about in your book is that we were doing this, and we were doing this without really the knowledge of natural selection before Darwin ever even got kind of wind of this. And so we've been doing this kind of artificial natural selection for a really long time, and so we can see that happening. So natural selection changes traits in a population in several ways. So we can have what's called a specific category, and that's where you've just got like two extreme phenotypes. And so the example they give here is a albino or a white tiger, and then an orange tiger. Or you can have something that's called a continuous range. Height is a really good example of this. Hair color, eye color, those are all, again, those are pleiotropic, meaning that there's multiple genes involved, and so that gives you that big range of sizes and colors and things like that. And so that's how we categorize those traits. It's either specific, so that's just like either one or the other, or continuous. So there's three different types of selection that we see happening naturally in populations. And so the first one here is directional selection. And that's when you've got one extreme phenotype that is that's favored over other phenotypes. And so for us, when we've selectively bred these little turkeys together, don't they look crazy? I like the wild turkeys that we have here in Missouri better than these big old white fluffy guys, but directional selection is going to give you one extreme phenotype. And so for the kangaroo rats, for example, the directional selection there would be to select for the little blonde rats, the little ones that are tan. And the selective pressure in that example is probably predation. And so the darker colored kangaroo rats are going to get picked off more easily and more quickly than the light brown ones. And so that's the selective pressure. In the example with the turkeys here that they've got up on the screen, the selective pressure is us, and we are selecting against the smaller, tinier breasted birds because we want to eat big fat turkeys with big fat breasts, right? That's what we want. And so rather than breeding together the smaller breasted turkeys, we're going to only breed together the turkeys that have the biggest breast muscles. And so after a few iterations of that population, you're going to have very big breasted turkeys that then you can have for dinner on your Thanksgiving or for just any other day when you feel like having Thanksgiving for dinner. So that's the selective pressure. And so again, directional selection, we are favoring one extreme phenotype. And so rather than having a range of these different phenotypes, we just select one. Stabilizing selection is when an intermediate phenotype is selected for the best. A really good example of this is birth weight in babies. I think we talked about this before, but having a really low weight baby is not good. They don't do very well, and they tend not to survive. And babies that are really big also tend not to survive very well, especially before the advent of modern medical intervention and technology. You can imagine somebody getting pregnant, you know, back in the 1500s with like a 12 or 13 pound baby. Chances are that baby's going to get stuck, and we're probably not going to be able to get that baby out in time. And mom and baby are both in danger then. And so the selective pressure is to have a baby that is not too big, not too little, but just right. Just like, oh gosh, I almost said Mary Poppins. What's that? Goldilocks and the Three Bears. We want it to be just right. And so little tiny babies, not so good. Really big babies, not so good. But between about 6.5 and 8.5 pounds, that's where you want to be in here. That's about the perfect size baby because it's small enough that we can get it out of mom without risking her health too much. And we can also have a baby that's big enough that it's going to be able to thermoregulate a little bit better, and it's going to do better when it's living on its own outside of the mother's body. So that's stabilizing selection. Disruptive selection is when you've got two extreme phenotypes and the intermediate phenotype has the lowest fitness. And so disruptive selection shows you here in this example, this is that sneaker males. I think I talked about these guys before too. And so relative size of male coho salmon here, like the really big ones, the females like to mate with them because they're like, oh, hey, look at that big daddy over there. Great, you're like giant and huge, and I want my sons to look like you, and so I would like you to mate with me. And so these ones are going to do very well because they're really big and the females like them, and they're all really excited to mate with a really big, awesome looking big male fish. And so the smaller fish, again, these are called sneaker males. The ones that are little itty bitty and they kind of look like females, those ones might not get selected for by the females, but the sneaker males can just kind of sneak in there, hence being called sneaker males, sneak in there and fertilize the eggs because they kind of look like females. And the females will allow this to happen because they think there's just another female coming over to say hello or whatever. And really she's like, oh my God, what are you doing? You're fertilizing my eggs, get out of here. And so they can fertilize the eggs before they're noticed. And that's how we've got the two populations of both larger and smaller males. The intermediate sized males are still recognizable as being male, but they are outcompeted for, and the females are not super excited or turned on, and they are not hot for those medium-sized males. And so this is where you see disruptive selection. Two extreme phenotypes have the best overall fitness. So natural selection can cause the evolution of what are called complex traits and behaviors. And so complex traits can be things like learning. And so fast learners, like in this example here that they've got like these little rats. Rats are actually very intelligent creatures. If you've ever had one as a pet, you probably can attest to that. And so what they did in this experiment was that they tried to breed and look for fast and slow learners. And so what they did here, the average number of errors committed here 220 or about 200 here. So after 6 generations of selection, what we did was we ended up with a whole bunch more slow learners, and then we had a whole bunch of fast learners down here. And so the rats that are making the fewest errors in the maze are selected for and bred with each other. And so after a while, you can start to see these traits kind of come out more and more as they're being selected for in these specific environments. Natural selection can also co-opt, or it can change the function of traits. And so the example they gave in your book was these insect wings. And so you might be wondering, like, how can something as complex as a insect wing evolve when it's such a complex thing, or like maybe an eyeball or something like that. And so what these experimenters did is that they had some flies that had tiny little wing nubs. These were not used for flying, but these were beneficial as far as thermoregulatory benefits were involved. And so they could like flap them around, and they could cool their blood down, or they could use them to just cool off. And so they were thermoregulating with these little nubs here, because they would just wave them around kind of like a diva fan, right? They've got 2 of them on their back. And so this was selected for, and so the ones that had bigger wing nubs for fanning themselves and acting like a diva were selected for. They got to breed more. So over the course of a few generations, what we end up with is bigger wing nubs. And so these are really good for fanning yourself, but now we also notice that these have some aerodynamic benefits. And so these are good for righting yourself when you're falling. Maybe we can fly a little bit, we can leap really far, and we can put those things out, and we can fly just like a little tiny bit. And so now we're starting to see a new function for this wing nub that we didn't have before, because it used to just be used for thermoregulation. And so after a few more generations, now we've got bugs that have these really big wings, and now they're used for flight and for aerodynamics, and they can still be used as a diva fan to cool yourself off. So put a star next to this slide. You guys are going to see this again. And so the fossil record and their lines of evidence, I want you guys to know the 5 primary lines of evidence for evolution. First one being the fossil record. Second one being biogeography. We talked about like the Pangea and the big supercontinent. When it's split up, we have these organisms that look very similar, but they end up on 2 different continents. And so again, that probably didn't happen by chance. What's more likely is that we had this supercontinent where we had tortoises that were all living together, but then as those continents kind of floated apart, you end up with tortoises in the Americas, and you end up with tortoises in Africa. Very similar organisms. They look different now because they've been apart for so long, but they're both still tortoises, right? So that's your biogeography. Comparative anatomy and embryology, so how these animals are formed. Molecular biology, so what does the DNA look like? What does the amino acid structure on proteins look like? How similar are we? And then also laboratory and field studies. So again, like I said, you can watch evolution happen in a petri dish. And so that's what we're doing here with the laboratory and field studies. We can actually watch it happen in a petri dish. So fossil record, this is our Archaeopteryx. You guys remember this guy. This is not actually the first feathered reptile, but it's the first one that we got a really good picture of here, and it's got wings. And it's also got feathers all the way down its back. And so the fossil record shows and it demonstrates the occurrence of evolution, because you can actually dig down through the layers and you can look at these fossilized remains of these animals, and you can actually see how they changed over evolutionary time. So fossils, for those of you that were unsure or unaware, are just preserved remains of organisms from long ago, like in Jurassic Park, right? We can use radio, like carbon dating, or we can use argon dating, because we look at isotopes, which are just different forms of an element, radioactive isotopes that break down at a constant rate. And so you can actually apply math to this. And when you look at these fossils, you can actually take out some of these radioactive isotopes, and we know how quickly they break down. And so if you look at how much of that compound is left, you can apply again an algorithm to it, and you can guess within a fairly narrow range how old that organism is. And so even if the law of superposition gets compromised, I suppose, like if we had an upheaval of like a mountain range or something like that, and then the fossils get all displaced, and so the oldest ones aren't on the bottom anymore, we can still use radio dating to kind of tell how old they are. And so in this case, they're using uranium-238. Its half-life is 4.5 billion years. Okay, so for half of that radioactive isotope to go away, it's going to take 4.5 billion years. And so if you discover some of this and you've got half of it left, you can kind of guesstimate how old it is based on where those isotopes are in their decay. So fossilization is not something that you see very often. There has to be a lot of things that go exactly right. So those animals had to get trapped somewhere where the bones weren't going to be degraded and eaten by something, so probably in an anoxic environment. There's only a few conditions where remains could be preserved. So maybe like in a tar pit, or if there's sap or something sticky, and again, maybe like an anoxic environment where breakdown is going to happen a lot more slowly, and so there's time for those bones to become mineralized and become rock. And only physical structures, or these are the only physical structures that can leave fossils. So kind of a cool thing. I was watching a documentary the other day where there was actually a cave lion cub that they found that was intact in this cave, and it was so cold and the air had so little oxygen in it that this cub that died like 10 or 15,000 years ago or something like that, pretty well preserved. It totally looks like it died a month ago. Still had hair and everything. You could see its facial features. Very cool. But again, very, 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 very infrequently will that happen. So we have a really well preserved fossil record for modern horses. And so this little guy back here, this Hyracotherium, is extinct, but we found fossils, and we can actually tell kind of when they went extinct, because we don't find the fossils in the shale or in the rocks anymore. And so then we start seeing these other organisms pop up that look very similar to Hyracotherium. And then over the course of time, so you can see down here, they just have time. They don't really have, measurable time on here, but just time. We've got Mesohippus, Myohippus, and we've got all these other horse-like organisms. And so the last extant species that was most like the modern horse today was this Merychippus right here. And so we found fossils of all these little guys, and we were able to kind of trace that back and see that Hyracotherium down here was probably the common ancestor for the modern horse. And again, they just change. Allele frequencies change, genotype frequencies change over time as the environment is changing, and as those genotypes and phenotypes become more or less advantageous in those environments. And so then we end up with a modern horse. Big. So let's see. This is a fossil that they found of a lobe-finned fish. This is called Tiktaalik, this guy down here. And so probably this is a missing link fossil between what are called the lobe-finned fishes. And so this is like a coelacanth fish. They actually have bones in their pectoral fins that act very much like legs, and early amphibians down here. And so we look at these homologous structures, these bones. You guys can see that they've got the same arm bones, phalangeal bones, wrist bones here. They've all got the same kind of setup. They look a little different because they were in different environments and probably used those for different functions, and they were separated by probably millions of years. But you can see that there are definitely similarities here. And so this shows us that we've got a link between, again, lobe-finned fishes giving rise to early amphibians. And so that's probably the common ancestry, or at least the link that we've got for the terrestrial amphibians that we see today. So again, another one of those examples that we see for evolution is that biogeography. And so biogeography, again, is where you've got an ancestral species that will either be the first to appear on an island. Island biogeography is very cool. Or we see similar species that appear on several different continents because they were probably one parent population at one point. Let me see how much battery I've got left. 23 minutes. Okay, we're good to go. So biogeography is just the distribution patterns of the living organisms around the world. And so the best place to study this, again, is the islands, because generally what happens with islands is as they come up out of the ocean, there's going to be one parent species that comes over, and then they don't really have any predators on islands because nothing has really come over yet, and then they can radiate. It's called adaptive radiation. They can radiate and they can speciate very quickly because there's no real competition for anything. And so honey creepers on Hawaii are pretty widely studied, as well as the finches on the Galapagos, because they're really widely adapted to a range of habitats, and they still look very much like their finch ancestor that is from 2,000 miles away. So probably what happened is we had a little population of finches that got blown over here. They were the first to arrive on the island, and because there was so much there to take advantage of, they were able to very quickly radiate into all those different niches. We've got marsupials and their placental counterparts, and so again, these guys are these are all a group of placental mammals, and then marsupials are not placental. They actually have babies and they put them in a pouch, just like koala bears and kangaroos. But we can see that we've got, again, this is from the Pangea thing, we've got Australian marsupials, and then we've got these little flying squirrels are in the United States, and so very similar organisms, but they appear on two different continents, and so we see that all over the place. Comparative embryology, so how these organisms grow in utero, and so you can see like all these vertebrate species have what are called pharyngeal gill slits, and they also have a post-anal bony tail at some point in their development. And so this is a human down here, so you can see that post-anal tail, pharyngeal gill slits, chicken, turtle, shark. So all of these have those similarities, and again, it's not likely that these just popped up separately from one another. In fact, that doesn't even really make any sense at all. What happened here is that we've got several organisms that as over time speciated, but they still have these similarities that are still seen in the embryological stage. Homologous structures, this is a big one. And so all of these animals here, we've got a porpoise, a bat, a horse, and a human. They all have the same arm bones. So they look a little different because they are adapted for different functions at this point, but we all share the same bone anatomy. And so again, that did not happen separately from one another. This happened over the course of a long time as these animals speciated, much more likely that we had a common ancestor and that we got these features from that common ancestor. Vestigial structures, this skull like freaks me out. Doesn't this look like alien? It looks like alien, like no eyeball because its eyeballs are pointing forward. Oops. So vestigial structures show us that there are animals that are still in the process of adapting because they've got structures that they used to be using somewhere in their ancestral history, but they're not anymore. And so one of the examples they give in your book is vampire bats having molars, even though they have a completely liquid diet. But their ancestors probably needed those molars, in fact did need those molars for eating things that were different than just a liquid diet. And so as time goes on, you're probably going to see those molars shrink and become totally extinct in this population because they're not needed anymore. It's just going to take a really long time for them to go away. So convergent evolution produces analogous structures, and so analogous meaning that they have the same function, but the ancestry where they came from is different. And so like this is a fruit bat and a dragonfly. They both have wings for flying, but the evolution of those structures came about completely differently. So that's kind of like the other side of homologous traits. Homologous traits have the same ancestry. Analogous traits have the same function, but different ancestry. Molecular biology, this is just us being able to look at DNA and proteins and patterns in proteins and the genetic code and looking at the similarity between different organisms. And this gives us gives us information on how related or not related two organisms are. So much so that you can actually apply time to this. And I think they have that, oh, here we go, the evolutionary clock. And so looking at hemoglobin, what they've got here is a human, a rhesus monkey, a dog, a bird, and a lamprey. And so they show all the similarities or the differences up here between the protein structure of hemoglobin between all these animals. And so you can actually apply math to this, and you can apply a timeline. And so 23 million years ago is when rhesus monkeys and humans probably first diverged as far as like our common ancestor went, and they can tell that because of the differences in the hemoglobin. And so you can do the same thing with dogs, birds, lampreys, and we are least related to the lamprey because we have the most difference in our hemoglobin protein structure. And so that's the evolutionary clock that they're talking about. Also, lab and field studies of looking at evolution happening like right in front of your eyes. So the best example that I can think of for this is antibiotic resistant bacteria. Bacteria that are exposed to antibiotics often and frequently. Again, bacteria love to mutate. They love to share notes with each other. They use those conjugation bridges with those sex pilots, and they share notes on those little circular pieces of DNA called plasmids. But once somebody figures out how to resist penicillin, they're going to very quickly share those notes. And then also, when you expose them to penicillin, all the ones that are not resistant die, and the only ones that are left are the ones that are resistant to penicillin. And so those are the ones that reproduce and make babies that have those genes for resistance. And so in the 1940s, almost all bacteria, oops, I keep doing that, almost all bacteria were not resistant to penicillin. But as we started using penicillin and exposing everybody to it, those bacteria started to show some advantageous mutations. In this case, we have resistance genes. And so over the course of time, and after we expose these guys to so much penicillin, what you have today is you have very few bacteria that are killed by penicillin because everybody has the news. Everybody's got the notes on how to resist penicillin. So again, this is happening all around us. And so when you hear somebody say like, I don't believe in evolution, probably what they're meaning is something different than what you're thinking. Because literally you can watch it happen in a petri dish. It's just like saying, I don't believe in books. That doesn't make any sense. You can see them. They are right here. Obviously, they do exist. And so if somebody tells you that they don't believe in evolution, they probably just have a misunderstanding of what that is. And so like we talked about in the beginning of this chapter with that fruit fly starvation experiment, remember when we first started out, the fruit flies couldn't go for very long without eating. I think it was like 4 hours. And after breeding together the ones that were able to go the longest, after 60 generations, they could go like 2.5 days or like something like that without food. And so that's natural selection and evolution. The population changed over time. That's all it is. And so with the fruit fly starvation experiment, it demonstrated that natural selection can produce dramatic changes in a population, and it can do so very quickly because as the environment is changing, if you don't change, you die. And then replicating that same evolutionary process can produce very predictable results. And so that's all that evolution is. So don't let it scare you. Don't get all freaked out and think that it's something crazy and mystical and magical, because it's not. But that's all that it is. All right, so that's the end of chapter 10. I hope you guys enjoyed this, and I will see you guys in class.